Hi, this is Miss Harkness, and today we are going to discuss how the cell cycle is controlled by internal and external mechanisms, and then we'll go on to discuss how the loss of this control of the cell cycle can lead to cancer. So let's get started. The process of cell division is regulated by a complex set of stages, and this is just to ensure that cells divide when necessary and then stop dividing when it's unnecessary for them to divide. So here on the left side of the screen we have human skin, which is composed of cells that are nearly constantly dividing and replenishing. On the bottom right you'll see a human liver, which has more regulated cell division, and that new cells are made only if the liver is damaged, so it needs repair, or if the liver is growing. And finally, at the top on the upper right side are nerve cells, which are specialized and do not divide in a mature human body, and also muscle cells fall under the same category as nerve cells. Alright, so in this diagram we have a more in-depth look at how this cell cycle is actually regulated via checkpoints. And the control system, which is shown in this diagram, is an operating set of molecules in a eukaryotic cell, and that just means that the cell has a nucleus, and the molecules trigger key events in the cell cycle, and the cell cycle is just the time from the birth, so from the start of the cell to when it's going to divide again. Okay, cells go through many stages during the cell cycle, the longest of which is interphase, and then they'll go on to mitosis and finally cytokinesis. Interphase is divided into three parts. Within the G1 phase, the cell grows. However, before it proceeds to the S phase, it must go through a checkpoint, which is shown by the star. And a checkpoint just decides whether the division process and the preparation for division should continue or if it needs to be stopped. And if conditions for the division are right, the checkpoint is passed, and then the cell enters the S phase, which is where the DNA is replicated. So that way when the information divides, each new cell will have all the genetic information that the parent cell had. And then the cell enters into the G2 phase, which is the final phase of interphase. And this is where the cell prepares for mitosis and continues to grow. And there is a G2 checkpoint, which is shown by the red star, that assesses the success of DNA replication before the cell enters into mitosis. Now during mitosis, the genetic information aligns in the cell, so the chromosomes align, and then fibers are attached to the chromosomes and they're pulled to each side of the cell, so that each of the new daughter cells will have the same genetic material, so they'll be identical to the parent cell. And during the process of mitosis and during the M phase, another checkpoint must be passed so that the cell completes the phases of mitosis and can continue to divide the rest of the cell, other than just the nucleus, which is divided in mitosis. And so that cell can continue on to cytokinesis, where the rest of the cell is divided, so you have the two daughter cells. Also in the cell control cycle are specific internal signals and external signals that provide stop and go signs at these cyclic checkpoints. So here we're going to talk about internal signals. Rhythmic fluctuations in the activity of cell cycle control molecules pace the events of the cell cycle and determine whether or not the phases occur and when they occur. So here we have kinase, which is a protein that activates and deactivates another protein and gives it the go-ahead signals at the G1 and G2 checkpoints. The kinases that drive these checkpoints must themselves be activated by the molecule cyclin, which is shown to the right, and is a protein that derives its name from cyclically fluctuating concentration in the cell. And because of this requirement, these kinases are called cyclin dependent. Alright, next we have MPF, or maturation promoting factor, which is involved in the stop and go signs at checkpoints internally as well. And MPF itself functions by phosphorylating key proteins in the process of mitosis. And the process starts when cyclins accumulate during the G1, S, and G2 phases of the cell cycle, and by the G2 checkpoint, enough cyclin is available to form MPF complexes, which initiate mitosis. Later in mitosis, MPF switches itself off by initiating a process which leads to the destruction and breakdown of cyclin. So this diagram, or the graph on the right side, shows how the concentration of MPF fluctuates and how the concentration of cyclin is directly related to that. And then the diagram on top shows how MPF is activated by cyclin and then later degrades cyclin when it doesn't need it anymore. So here we have external signals that affect the control of the cell cycle, and PDGF, or platelet-derived growth factor, represents an external signal that is required for the division of fibroblasts, which are essential in wound healing. And when an injury occurs, platelets, which are blood cells that are important for blood clotting, release PDGF, and fibroblasts are connective tissue that possess PDGF receptors on their plasma membranes. So when the PDGF binds to these receptors, that activates a pathway that leads to the production of fibroblasts and a healing of the wound. So given the information that we know about cell control cycles, in normal cells it would be expected that stop signs would be signaled after the cell density reached a given amount, and this concept is called density-dependent inhibition. So the problem occurs when there are errors at the checkpoints and cell division becomes uncontrolled, where the cells are not affected by factors that normal cells would be affected by. And so on the right we have cancer cells, which you can see are growing in multiple layers, whereas the normal cells on the left stop growing after they reach their density. So that leads us up to cancer, and cancer in simplest terms is uncontrolled cell growth. And so here you have a picture of cancer cells, and cancer cells are problematic because they don't respond to the body's control system, and therefore they can divide excessively and can invade other tissues, and they can also grow on top of one another in layers, like we just talked about with the density-dependent inhibition, so that doesn't apply to them. And cancer cell growth stages are also irregular, so if growth halts, it is at a random stage in the cycle. And finally, cancer cells can divide indefinitely if the correct nutrients are present, and that's why cancer is one of the major problems that scientists and doctors face today. So on the right you have a picture of a tumor, which is just showing uncontrolled cell growth. Thanks for watching. I hope that was helpful.